Good afternoon, dear student. We can start the lecture. Enzymes, structure and properties, the mechanism of action. The purposes of this lecture: biological catalysis, definition, studying the features of structure and function of enzymes, finding out the principle of nomenclature and classification of enzymes, and we should get acquainted with marker and organ-specific enzymes. The content of our lecture: general characteristics of enzymes. Later we shall discuss the historical background of enzymology, enzymes, their chemical nature, structural organization of enzymes, properties of enzymes, mechanism of enzyme action. So what is the enzyme? Why they are so important for medical students? Life depends on the existence of powerful and specific catalysts, the enzymes. Almost every biochemical reaction is catalyzed by an enzyme. With the exception of a few catalytic RNAs, all known enzymes are proteins. Many require non-protein coenzymes or cofactors for their catalytic function. Enzymes are classified according to the type of reaction they catalyze, and all enzymes have formal enzyme classification number and names, and most have trivial names. General characteristics of enzymes. Enzymes are biochemical catalysts. Enzyme is a protein molecule that speeds up chemical reaction in all living things. So these are the most often used terms for enzyme. Without enzymes, this reaction would occur too slowly or not at all, and no life would be possible. And the word enzyme itself derived from Greek word meaning in yeast. Many scientists who studied enzymes in 18th or in 19th century studied reaction catalyzed by enzyme in yeast. So later we shall discuss historical background, properties of enzyme, and structure of enzyme. Short historical background of enzymology. In ancient times, people used enzymes in various technologies: bakery, winemaking, processing of height, and others. In 18th century, Reaumur and Spallanzani described digestion in birds. Later, in 1814, Kirchhoff showed the characteristic of catalytic hydrolysis of starch during grain germination. 1833, in this year, French biologists Payen and Persaud isolated a malt-soluble ferment or enzyme and called it diastase. 1835, John Berzelius, he coined the term catalysis. In 1850s-60s, Louis Pasteur stated there is no fermentation in heated flask. In the middle of 19th century, it was debates between Eustace Liebig and Louis Pasteur about organized and unorganized enzymes. 1871, in this year, Manassene, and then 24 years later, Buchner showed that cell extract capable of catalyzing. So not always enzyme should occur with the cell. So also the catalysis can be performed without cell. They grinded yeast with sand, filtered it, and then observed fermentation. 1878, Wilhelm Kühne coined the term enzyme. 1894, Emil Fischer created lock and key model. And 1903, Victor Henry stated that all enzymes are proteins. Realization of enzyme nature. 1913, Michaelis and Menten created the theory of enzymatic catalysis. Then in 1929, Sumner demonstrated the protein nature of enzymes, urease and others. He used the enzyme extract from beans. Later in 1933, the primary structure of RNAs was studied. 1968, Merrifield, artificial synthesis of RNAs was performed. And nowadays, the work are on synthesis of artificial enzymes. Enzymes, chemical nature, structural organization of enzymes. Structure of enzyme. Enzymes can be divided into simple or conjugated enzymes. Simple consists of amino acids only. Conjugated contains also non-proteinous part, organic or inorganic molecule known as a cofactor. Cofactor can be metal ions or organic molecule referred to as coenzyme. Coenzymes are non-covalently attached to enzyme. Many coenzymes have vitamins or contain vitamin as a part of their structure. On the other hand, prosthetic groups covalently attached to an enzyme. So coenzyme usually actively involved in the catalytic reaction of the enzyme, often serving as intermediate carriers of functional group in the conversion of substrate to product. The catalytically active complex of protein and prosthetic group is called the holoenzyme. The protein without the prosthetic group is called the apoenzyme. It is catalytically inactive. So metal ions and some enzymes can occur together. So metal ions like iron, copper, zinc or magnesium can be found in enzyme listed in this table. So I can mention cytochrome oxidase, catalase, peroxidase, which depend on iron. Cytochrome oxidase also contain copper, 2+. So this enzyme, copper and iron, they can change their valency. So that is why they are very good used for transportation of electrons. Zinc we can find in DNA polymerase, carbonic anhydrase, alcohol dehydrogenase, while magnesium can be found in hexokinase, glucose 6-phosphatase. Manganese is often found in arginase, while potassium in pyruvate kinase, which also requires magnesium. Nickel in urease, molybdenum in nitrate reductase in plants, but selenium in glutathione peroxidase. Coenzymes can serve as transient carriers of specific atoms or functional groups, like thymine pyrophosphate, termed as TPP, is a transfer group aldehyde, so it can be found in pyruvate dehydrogenase. Flavinadenine dinucleotide, FAD or FAD, transporter of hydrogen atoms, enzyme which contains this is succinate dehydrogenase. Nicotinamide adenine dinucleotide, NAD, is transporter of hydride ion in alcohol dehydrogenase as an example. Coenzyme A, transporter of ethyl group, acetyl-CoA carboxylase. Pyridoxal phosphate or PLP is transporter of amino group. Example, aspartate aminotransferase. Vitamin B12 is a precursor of, is a form of enzyme coenzyme which transport hydrogen and alkyl group, methylmalonyl coenzyme amylase. Carboxyl group can be attached to substrate with the help of biotin. So biotin is a prosthetic group because of covalently bound to enzyme and can be released in the form of biocytin. Propionyl coenzyme carboxylase is an example. Tetrahydrofolic acid, transport of one carbon group other than B12. Simidulate synthase is an example of this enzyme. Some coenzyme structure is listed in this slide. So you can see the structure of vitamin B1 thiamine, riboflavin B2, pantothenic acid B3, nicotinic acid and nicotinamide, which are termed as vitamin PP. Pyridoxine, which is vitamin B6, can be found in 3 forms. Also, you can see coenzyme form of vitamin B1 is thiamine pyrophosphate. Pyridoxal is pyridoxal phosphate. So we should also know the structure of coenzyme Q, which is necessary for bioenergetic. Also, ATP is not coenzyme, but is a transporter of high energy bonds. ATP converted to ADP, AMP, and cyclic AMP is a transporter of signal, transducer of signals. Some coenzymes of dehydrogenases derived from pyridinic and flavinic coenzyme. Pyridinic is NAD or NADP, and flavic or flavinic FMN or FADH. The structure of NAD, NADH, and NADP, NADPH are shown in this picture. In the lower row, you can see the structure of FAD, FADH2, FMN, and FMNH2. So some enzymes can consist of several proteins. Monomeric enzymes are made of a single polypeptide, while oligomeric enzymes possess more than one polypeptide subunit. Certain multi-enzyme complexes possess specific site to catalyze different reactions in a sequence. The specificity of enzyme is dependent on the APO enzyme. In the next picture, we will see the structure of enzyme. So we can find active site of the enzyme, which is defined as a small region at which the ligand, substrate, or regulatory molecules bind and participate in catalysis. Substrate binding active site includes binding site, bind and orient substrate, catalytic site, convert substrate to product, and releases them. Also, some enzymes possess allosteric regulatory site, like sites which can bind activators or inhibitors. So some enzymes which has this regulatory regions or active sites are called allosteric enzymes, allosteric site with allosteric enzyme. So these enzymes are allosteric, and the activity of this enzyme depends on the factor of the enzyme, modulators of their activity, positive or stimulatory effectors and negative or inhibitory effectors. Allosteric effectors catalyze key regulatory steps in biochemical pathways. Next moment, properties of enzymes. There are 10 salient features of active site. First, depend on native 3D structure of the enzyme. Second, made up of amino acids, catalytic residues, which are far from each other in linear form but close to each other in 3D form. Exist as a cleft or crevice or pocket occupying a small region, rather flexible, possess a substrate binding site and catalytic site. Cofactors can be part of this. Substrate bind by weak non-covalent bond, determines enzyme specificity. Also, active site can contain repeatedly amino acids: serine, aspartate, histidine, cysteine, lysine, arginine, glutamate, tyrosine, etc. Enzyme substrate complex is formed and product can be released. So now we shall discuss features of enzymatic catalysis. Like any catalysis, organic or inorganic, enzymes neither consume it nor produce it during the course of their reaction. Do not cause the reaction to take place. Do not alter the free energies of substrate or the product. Alter the rate but not the equilibrium constant of reaction. But there are some differences. Enzymes are proteins. Enzymes are more effective because they can act in very small concentration, very small amount. They are highly specific. They function within a moderate pH and temperature, compared with non-organic catalysts, which require high energy or very low or very high pH. Enzymes are capable of being regulated. So let's see some factors which can influence enzyme activity. So these factors are temperature, pH, concentration of enzyme, concentration of substrate. So what about concentration of, what about the temperature? Increase of temperature can also increase enzyme activity, but to a certain extent. When denaturation of enzyme occurs, after this activity of enzyme will drop. pH. Every enzyme has its optimal pH, and this is a rather narrow region when enzyme is active. So left to right, so lower or higher pH, and then activity will drop drastically. Enzyme concentration will increase velocity of enzyme reaction in linear manner, while substrate concentration result in increase of velocity in hyperbolic manner. So later we shall discuss the feature of enzyme kinetic, and these parameters which are shown here are Km, Michaelis constant, and 1/2 of V maximal will be described and explained later. Now let's say about enzyme nomenclature. So there are some enzymes which have trivial names like pepsin, trypsin. Later it was proposed rational names. The adding of suffix "ase" to substrate result in the name of enzyme. For example, urea, urease; amylose, which is a starch, amylase; glucose 6-phosphate, suffix "ase", glucose 6-phosphatase. So rational nomenclature, but this nomenclature was not very good, because later in 1961. Systematic naming was proposed by International Union of Biochemistry. It was stated 6 major classes of enzymes, depending on the reaction they catalyze. Class 1 oxidoreductases, class 2 transferases, third class hydrolases, fourth class lyases, fifth class isomerases, and sixth class ligases or synthetases. And each class has a code number, EC. 1 is a class, 2 is subclass, 3 is subclass, and 4 is specific enzyme in the list. Enzymes can be named by the reaction by the type of reaction they catalyze. So oxidoreductases usually transfer the electron, hydrogen, and hydride ions. Transferases transfer a functional group, for example, ethyl group. Hydrolases usually cleave bonds between COC and CSO by adding water. Lyases add groups to or from double bonds. Isomerases transfer group within the same molecule, while ligases join molecules nucleotides with the help of ATP as a donor of energy. But now you should know 7th class was added in 2018. So now this nomenclature is updated. The 7th class is translocases. Translocases which transfer substances from one site divided by membrane to another, from one compartment to another. The mechanism of enzyme action. To discuss this moment, we should remember some terms from thermodynamics. Energy change occurring during the reaction are shown in this picture from Lippincott textbook. Chemical reactions have an energy barrier separating the reactant and product. This barrier is called the free energy of activation, which is the energy difference between that of reactant and the high energy intermediate that occurs during the formation of product. So the change in energy during the conversion of molecule to reactant or reactant A to product B is processed through the transition state or high energy intermediate, which is termed as T asterisk. So reactant, transition state, then product B, A to B. So free energy of activation is a peak of energy, which is the difference for reaction catalyzed or uncatalyzed. In the picture you can see the difference. For activated or catalyzed reaction, this reaction will be lower. So that is why enzymatic reaction will run much faster. Energy diagram for the reaction catalyzed by chymotrypsin, for example. So you can see enzyme catalyzed reaction occur with several peaks, and these peaks correspond to different state of transition state. So the energy barrier to the transition state is lowered in the enzyme catalyzed reaction by the formation of additional bond between the substrate and enzyme in the transition state complex. The energy is provided by substrate binding to an enzyme. The enzyme does not, however, change the energy level of substrate or product. So transition state intermediate you can see in this reaction. In non-catalyzed reaction is like one-step reaction, S to P, substrate to product, while enzyme catalyzed reaction is divided in several steps. Enzyme plus substrate is the initial moment, then enzyme substrate complex forms, then enzyme substrate 1 asterisk, enzyme substrate 2 asterisk, enzyme substrate 3 asterisk, and finally enzyme and product, which are released from the active site. So in spite of this multi-step reaction, the total reaction will occur much faster because the energy of activation is lower. So here is the example of the active site of enzyme, and you can see several amino acid residues which join together to assist catalytic reaction. So you can see non-polar pocket, which is formed by several nonpolar amino acid, which can find hydrophobic structure radical of tyrosine, for example. Yes. Then we can see histidine in 69th position, in 196th position, which are joined together. Glutamate 72nd position. So this three amino acids bind zinc. Then glutamate, tyrosine, arginine. So all these amino acids are interacting with substrate and make possible hydrolysis of the bond between tyrosine and other amino acids which are bound with peptide bond. So what are the explanation how enzyme can catalyze all the reactions? There were two theories proposed by Emil Fischer and Daniel Koshland. Emil Fischer proposed this theory in 1894, and he called this theory the lock and key model. Later in 1958, the induced fit or glove and hand model was suggested by Daniel Koshland. So what are the difference? So Emil Fischer supposed that the perfect binding of specific substrate to specific enzyme is like one key to one lock. So coincide like one key to one lock. So, but it was good for highly specific enzyme. But what about enzyme with low specificity, which can act on different sizes? So for this, Daniel Koshland theory was much more convenient. Unlike the lock and key model, this model shows that enzymes are rather flexible structures, and they exercise constantly reshape by its interaction with the substrate until the time the substrate is completely bound to it. So in other words, we can say in lock and key model proposed by Emil Fischer, both substrate and enzyme are rigid structures. In the second model, induced fit model, substrate is still rigid, but enzyme no more. Enzyme is flexible. According to the modern views, both both structures are flexible, substrate and enzyme. Effect of substrate concentration of reaction on reaction velocity. So now it's time to explain kinetic of enzyme reaction. So you remember that this plot is hyperbolic. Maximal velocity. So enzymes which follow Michaelis-Menten kinetic are shown in this plot. Maximal velocity is the rate of an enzyme catalyzed reaction increases with substrate concentration until a maximal velocity is reached. But the leaving off the reaction rate is a high substrate concentration reflects the saturation with substrate of all available binding site of the enzyme molecule present. But you see, this value is very difficult to define because it's asymptotic value. What does it mean? So the more concentration of substrate, the more will be velocity maximal. We cannot say definitely which substrate concentration is necessary to define V maximal. So hyperbolic shape of the enzyme, kinetic curve. So some enzymes can show, like allosteric enzyme, sigmoid curve. But now let's pay more attention to Michaelis-Menten enzyme. Most enzymes show Michaelis-Menten kinetic, in which the plot of initial reaction velocity against substrate concentration is hyperbolic. But in contrast, allosteric enzymes do not follow Michaelis-Menten kinetic and show sigmoidal curve. So effect of substrate concentration of reaction velocities can be described by Michaelis-Menten equation. So enzyme 1 with small Km and enzyme 2 with large Km. So Km is constant, like Vmax is also constant. And the argument is substrate, concentration of substrate. So you can see this reaction, and this equation describes hyperbolic plot. So the higher Km, the lower will be enzyme velocity of enzyme reaction. So Km show enzyme affinity to substrate. So we can find several regions of this plot. In the beginning, at low concentration of substrate, when concentration of substrate much less than Michaelis constant, the velocity of the reaction is approximately proportional to the substrate concentration. But when high concentration of substrate and the velocity of substrate reaction is closer to V maximal, so begin a reaction of zero order. The velocity of the reaction is zero order. That is the constant and independent of substrate concentration. So hyperbola versus straight line. So due to this hyperbolic reaction and inability to calculate V maximal, some scientists, chemists proposed how to transform this equation into linear form. And this was proposed by Lineweaver and Burke. Michaelis-Menten equation can be converted to linear form by transformation of hyperbolic equation to linear. So Lineweaver and Burke proposed this transformation, and now you see is a linear equation. Double reciprocal plot. This plot derived from a linear transformation of Michaelis-Menten equation. So according to this Lineweaver-Burke plot, we can see and calculate the maximal velocity of enzyme and also Michaelis constant. The original reason for this sort of transformation was to calculate Km with Vmax from experimental data. Nowadays, these are computer programs that can accurately feed the data to hyperbolic curve and calculate the constant, so the Lineweaver-Burke plot is no longer necessary for this type of analysis. The Lineweaver-Burke plot can illustrate some general feature of enzyme kinetics. Now you see not Km but -1 Km on the horizontal axis, 1 divided by concentration of substrate, and on the vertical axis we can find 1 divided by Vmax. So this is the main characteristic of enzyme. So now some words about enzyme specificity. Enzyme specificity can be of several types: substrate specificity, reaction specificity, or stereospecificity. Substrate specificity can be absolute, relative, and broad. Relative can be group specificity or bond specificity, while stereospecificity is distinguishing optical stereoisomer L amino acid and D amino acid oxidases. Reaction specificity: amino acid can perform transamination reaction, oxidative deamination, decarboxylation, and other reactions. And substrate specificity: absolute, for example, urea cleaves only urea, glucokinase recognizes only glucose. And relative: trypsin hydrolyzes peptide bonds at positive radicals of amino acid, arginine, lysine, while chymotrypsin recognizes aromatic phenylalanine, tyrosine, and tryptophan. Or bond specificity: glycosidases, glycosidic bonds, lipases, ester bond, and lipid. Broad specificity are hexokinase, acts on glucose, mannose, and glucosamine, but not on galactose. The ranges of enzyme specificity. So we can place this ranges of enzyme specificity according to this table. Decreasing specificity: the highest specificity is absolute or unique substrate specificity, urea, then stereochemical, then group or function specificity, and finally low specificity. How to measure what are the units of enzyme activity measurement? The International Commission of Enzyme defines one international unit of enzyme as the amount that catalyzes the formation of 1 micromole of product in 1 minute. Because enzymes are very sensitive to factors like pH, temperature, and ionic strength, the conditions over time must be specified. Another definition of unit is the unit catal. 1 catal is the amount of enzyme catalyzing the conversion of 1 mole of substrate per 1 second. But this unit is very high, and usually enzyme activity can be expressed in derivative. Specific enzyme activity can be express it in micromole of substrate per minute per milligram of enzyme. And also turnover number is the number of substrate molecule metabolized per enzyme molecule per unit of time with unit of minutes of seconds. So this is the values of turnover number for some enzyme. The most active enzyme is catalase, 40 million catalytic reaction per second. Next one is carbonic anhydrase, 1 million reaction per second. Acetylcholinesterase is rather fast enzyme. Penicillinase, 2000 reactions per second. Lactate dehydrogenase, 1000. Chymotrypsin, 100 reactions. DNA polymerase, 15 reactions per second. And lysozyme, only 0.5 reaction, 1 reaction, 2 seconds. So let's state some conclusions. Most enzymes are proteins by their chemical structure. Enzymes are extremely effective catalysts. They have some advanced features comparing with inorganic catalysts. Enzyme activity depends mostly on temperature, pH, enzyme substrate concentration. And enzyme activity can be expressed in international units, catalysts, and other units. Enzymes are important markers of pathology, so they are widely used in medical practice. So, also in conclusion, I can recommend you to study this key concept map for the enzyme, which explains you enzyme reactions of enzymes and the rate of enzymatic reaction. So these are the sources of which were used for preparing to the lecture. So the lecture is over. Thank you for your attention.